These are the notes on DNA, and excuse me, let me get the screen set up. But DNA stands for deoxyribonucleic acid, and if we look at just why we study DNA, lots of important reasons. Number one, we study DNA to find out all the different genetic traits in humans. The Human Genome Project, which was completed in 2000. It was a study that mapped out all the human genes, and we, as scientists, did not understand or know every gene in the human race up until about 2000. We also use or study DNA for identification, whether that's something like a crime scene investigation or a maternity or paternity cases. It's definitely used. And we leave DNA all over the place. Cloning, whenever we clone animals or clone cells, gene therapy. A lot of our food is genetically engineered. Vaccines are genetically engineered. Drugs are genetically engineered. Some reagents that we use. So lots of reasons why uh, individuals need to study DNA. All very helpful things in our society. Um, and without this groundbreaking work from some of these scientists, we would not be able to use some of these things that we see on this slide. So the first guy that we want to talk about is Frederick Meacher. Extracted white substance from the nucleus of cells. He called it a nucleic acid. He was the first one to sort of get this idea of what uh, DNA was. Chargaff found in that nucleic acid, in that substance, that white substance, that there were some nitrogen bases that ended up pairing up with one another. That's why we call them the Chargaff rules. And that was 1949. So we had this gap here between these years where we didn't really find anything. And 1949, Chargaff found this, and so you can see that it's still pretty recent that we found this information about DNA. And here are the big ones. Hershey and Chase in 1952 worked with viruses and bacteria and mice and proved that DNA, in fact, was the genetic information or genetic material. Um, and then Rosalind Franklin and Maurice Wilkins. And we nowadays talk more about Rosalind Franklin and her contributions because without her studies, Watson and Crick are definitely not even famous. Maurice Wilkins actually took a lot of Rosalind Franklin's work and handed it over to Watson and Crick. And Watson and Crick then developed their 3D model or their shape of DNA and became famous. And Rosalind Franklin did not. And in fact, that a lot of her work, she actually worked in a radiation lab, X-ray, a diffraction lab, and in that process in her life's work, um, she got cancer as a result and ended up dying as Watson and Crick were becoming famous with their model. But uh, needless to say, it was her photo 51 X-ray diffraction that Maurice Wilkins actually, again, kind of took from her that enabled Watson and Crick to develop their 3D model. Chances are that she probably would have um, been the one that developed this model had she not died of cancer. So let's look at what DNA is. It's deoxyribonucleic acid. Um, it's the blueprint for all the proteins in our body. We often call this the genetic code, but it's simply just the genetic code to make proteins. And we'll get into more of the structure later on. If you want to look at what it looks like here on the left-hand side over here, you're actually going to do this in the lab today. We're going to look at banana DNA, but it looks like white fuzzy stuff. And then under a nanoscopic view, we can see that double helix where we have bases that are pairing up in the middle. DNA size is really small, about 6 micrometers in diameter, but in each cell, that's about 1.8 meters. So think about that. 1.8 meters of DNA per cell, and you're made up of billions of cells. So if you did the calculations, you could probably go to the moon and back with just your own DNA if we wanted to stretch that out, stretch out all those chromosomes. So if we look at the structure of DNA on the second page of notes, DNA is composed of subunits called nucleotides. If we think all the way back to chapter 2, this is one of those macromolecules or organic molecules that we studied earlier. And uh, the nucleotide is that base unit or that subunit. There's three parts to that nucleotide. You have the bases, you have the deoxyribose, the sugar, move that out of the way, and you have the phosphate group. And all three of those work together. You stack a whole bunch of these nucleotides up together and you get your double helix DNA molecule. The four bases that are involved, and this is just like a puzzle, and actually you're going to be building the molecule today as well. Adenine, thymine, cytosine, and guanine. These are just nitrogenous bases that actually pair up, just like in this slide. You have a little puzzle. A and P look like they, A and T, I'm sorry, look like they can go together, and C and G look like they could go together as well. And this is the code. These base pairs in the middle is the code to make our proteins. This is what gives us our traits. The code to make the proteins that give us our traits. So it's these four letter combinations are extremely important in determining what we look like and who we are. These base pairs are called complementary base pairs because they match up with one another. Now, taking a look at the double helix, as we see, we have looks like a twisted ladder pattern where you have the rings in the middle and the rungs on the side. And we look at the sides or the sides of the ladder. We have the sugar in red and the phosphate groups, which is the little circle there that makes up the sides of the ladder. And then the rungs, the bases, the A pairs with T and C pairs with G, and that is the code. And that's held together by hydrogen bonds. But the most important thing is that code there that's going to make our proteins. So if you get a chance, go ahead and in your notes just copy the strand. If this was one strand, what would the other side be? A pairs with T, G pairs with C, G pairs with C, and so on. So I'm not going to take any more time in doing this. We'll get much practice throughout the day today. If we want to look at DNA replication, because it's the next piece that you really have to understand. DNA replication means to make a copy, and we've used that term copy in the S phase that we just talked about in the cell cycle. If we want to make a clone of cells, then that DNA has to be copied first. So what is this? So precisely what the slide says, what does this sound like? It sounds like mitosis, just what we talked about, actually more the cell cycle. Before mitosis can happen, the DNA must be copied in the S phase of the cell cycle. And this, of course, happens in the nucleus of cells, and it happens before cell division. So it's really, we're kind of putting the pieces together here by talking about DNA replication. There's three steps to the DNA replication. It's pretty basic. The DNA molecule is going to unzip the new base pairs that are floating around in the cytoplasm, pair up, and then the DNA is going to be proofread. There's a molecule or an enzyme that actually does that. So if we look at the first step, DNA replication, the molecule is going to unzip by an enzyme called DNA helicase. And again, anything with the word ASE on it is going to be an enzyme. So that enzyme just kind of unzips it just like you would unzip a code. It's going to unwind that molecule, breaking apart those hydrogen bonds. And so you have all these empty spaces on each side of the template. And so the nitrogenous bases that are floating around in the cytoplasm are going to come in and match up with their, come into the nucleus, that is, and match up with their base pair. A pairs with T, C pairs with G until we have two identical strands of DNA made. There is an enzyme called DNA polymerase that is going to help add the bases together. It's just going to be a helper. And the last part is to proofread the molecule. It's really important that we proofread the molecule because anything that is mismatch can create mutations that can occur. And as we've talked about throughout this year, some mutations can be pretty harmful. Can lead to things like cancer and other genetic issues. But during proofreading, the DNA polymerase removes any mismatched bases That's part of its job as being a helper, is just double checking to make sure that A is pairing with T and C is pairing with G. But sometimes things do happen. For instance, in E. coli, which is a prokaryote, a bacteria, it's about one base in every 1 million are going to be in air. And after proofreading, the error rate is about 1 in a billion base pairs. And that's just E. coli. So think about how sophisticated humans are. Mutations do happen, though. These are the permanent changes in the DNA code, something that the DNA polymerase does not catch. And how does the DNA get damaged, or how does it mutate? Well, radiation, UV rays from the sun, X-rays, radioactive elements, uh, chemicals like pesticides or cigarette smoke can all damage your DNA. In fact, we walk outside and our DNA is being damaged constantly by the sun. That's why it's so important to wear sunscreen, no matter how your skin reacts to it, whether you get burned or whether you tan. It's still really important that you wear sunscreen out there. These things that cause DNA to be damaged is called a mutagen. Anything that causes a mutation. So that about has it with our DNA notes. I do want to show you just briefly how DNA replication works, so you sort of have a visual here. If this is our DNA molecule right here, and we want to copy that, DNA helicases are going to unzip it. DNA polymerase is going to make sure that these bases are matched up on the right side, and if there's any mistakes, it's going to kick out a nucleotide and bring in the right one. So here it goes in a really fast order. There we go. And you see C pairs with G and A pairs with T. And then the result, of course, are two identical strands. This concludes the end of the DNA replication notes. Make sure you have these in your notes and prepare for the lab.